Hi everyone, my name is Blanca Marisol Dominguez Martinez, and today I'm pleased to present our work entitled Evaluation of Anthocyanins in Polymeric Matrices for 4D Printing Applied to Food Safety. This research was conducted at the Tecnológico Nacional de México, Campus Querétaro, in collaboration with the Universidad del Valle de México. The motivation behind this work is the growing need for intelligent materials capable of monitoring food quality and safety. Anthocyanins are natural pigments that change color according to pH variation, making them promising candidates for smart sensing applications. For this reason, our objective was to evaluate different plant sources of anthocyanins, characteristics, and incorporate them into polycaprolactone or PCL for potential use in 4D printing for food safety. Also, anthocyanins are widely distributed natural pigments found in fruits, vegetables, and flowers. Besides their antioxidant properties, they exhibit a remarkable color response to pH change. Because food spoilage is often associated with change in acidity, anthocyanins can act as a natural indicator capable of providing visual information about food freshness. Therefore, the main objective of this work was to extract, compare, characterize, and apply anthocyanins from different plants sourced into polymeric matrices suitable for filaments production and 4D printing application. The evaluated sources include raspberry, blackberry, hibiscus, purple cabbage, beetroot, lucorn, poicentia, and tradescantia pallida. In this moment, I'm going to explain the methodology. For the first stage of the investigation, we select the different plant materials and vegetable sources like red cabbage. The A is the Tradescantia pallida, hibiscus is the B. We choose only 5 red cabbage and blackberry, strawberry, and these different plants, we extract anthocyanins for different techniques of extraction. We use 3 different techniques. So was use ultrasound in hibiscus and red cabbage too, and let me see, let me zoom this. It's better. We extract with ultrasound probe and VAT too, and controlled heating too, and with the pulse electric files like blackberry, strawberry, and red cabbage too. The extraction process employs ethanol and acidified solution in order to maximize pigment recovery and stability. After extraction, the stage 3 was evaluation the change color, modifying the pH of the solution and showing this change color in every extracts. In the stage 4, we character of extracts with the Fourier transform infrared spectroscopy or TER, and we use two UV visible spectroscopy, and we make a change for the color. Finally, we impregnate of extracts in polycaprolactona, PCLs, and we made a filament for printed in the 3D printed with the Creality printed, and we use into a pencil. Finally, we obtained the PCL and anthocyanin samples. The material was colorful, and for the results, now I will discuss the main results. First, we show the principal change with the pH measure for Pocentia, hibiscus, purple cabbage, beetroot, and Tradescantia pallida. One of the most important observations in this stage was the clear color responsive of the stretch of pH change. The pH scales obtained demonstrate the several extracts exhibit noticeable chromatic transition between acid and alkaline environments. This confirms their potential and visual indicator, like we show in the figure 7. Among the evaluated samples, blackberry extract showing particularly strong and evident color change. For this reason, this is the printing material changing with the pH, pH acidic, neutral, and alkaline. Blackberry acidic, neutral, and alkaline in the printed test pieces. This was after incorporation into the PCL matrix and subsequently printing. The produced specimens retain part of this chromatic functionality. The printed structure exhibit visible color difference, demonstrating that anthocyanins can remain active after processing. These findings support the possibility of develop smart print materials capable of signal environment of food quality change through this color variation. For the results with the FTER control spectra, we obtained first the controls. We use 3 controls: water, obviously, cyanidin for sigma, and blue corn anthocyanins. This anthocyanin was provided for another laboratory lab, and in this case, the anthocyanins of blue corns was purified. So in these 2 patterns, we can find the signal of the anthocyanins. So regarding chemical characterization, FTER analysis provide evidence of the successful extraction of anthocyanin-related compounds. We found in this table the vibrational assignments, especially we show them 1,380 to 1,320. The phenolic ring vibration compounds and lysyanidin. We have in cabbage, hibiscus, blackberry, and raspberry. We found in the 1,250 and 1,150 flavonoids and compound glycosides in red cabbage, hibiscus, blackberry, raspberry. And in all samples, we show sugar-linked anthocyanins in all samples and all extracts in the 1,100, and 1,000. And we found two in the 500 and 800 compounds about aromatic ring deformation, like in hibiscus, red cabbage, blackberry, and raspberry. Finally, UV visible spectroscopy also confirmed the presence of target compounds, blackberry and strawberry extracts, exhibiting two characteristic absorption maxima. We found 280 and 283 nanometers associated with phenolics, compounds, and flavonoids. And in the other, we found 535 and 138 nanometers corresponding to anthocyanins pigments. Together, these results validate that in the materials, these results validate the effectiveness in the extraction process and they confirm the presence of anthocyanin in extracts and also in the printed material. Finally, I was ending with the conclusion. Blackberry showed good impregnation in PCl and evident color change. Raspberry and blue corn produce faint tones attributed to their sugar content with affecting printing. Ponce require higher concentration to achieve a noticeable chromatic change. FDER expects comparison with cyanide and blue corn anthocyanin standard confirmed the presence of anthocyanin-related functional groups. Ultrasound and PEF treatment preserve these characteristic bands more effectively than conventional heat treatment suggests the ancient extraction. And in retention of anthocyanins and polyphenolic compounds. The UV spectra and blackberry and strawberry extracts displaying two characteristics: absorption maxima, an intense band, and an ultraviolet region, attributing to phenolic compounds and flavonoids, and a second band visible region corresponding to anthocyanin pigments. Finally, I want to thank you very much for your attention. This work contributes to the development of sustainable and intelligent materials based on natural pigment, opening new opportunities for smart food packages and food safety monitoring through 4D printing technologies. I will be happy to answer any question in my email here in the form in the abstract of my work. Thank you very much.